So welcome back to Chapter 1, Part 2, and this is where we're actually going to start to delve into trying to figure out how to answer that causal question that I left you with at the end of Part 1 of Chapter 1, and that was, why do female honeybees mate with so many males? Remember that that's a causal question. All why questions are causal questions. And so, um, that's what we're going to try to answer, and of course we answer it with a hypothesis or many hypotheses. With the E there, that's plural for hypothesis. So recall that I've already defined this for you on a previous slide, and a hypothesis is a possible explanation. So I talked about how it should be specific. Remember I said it should be narrow, and they're kind of equivalents. And it also should be testable. And so when you're thinking about a possible explanation for why female honeybees mate with so many males, you should keep in mind that maybe you as an individual, maybe you can't figure out how to test it. We'll work on that later. But just try to come up with a couple of reasons. I'm asking you for 3. Go ahead and pause this video, get out a piece of paper, write down your 3 hypotheses. It might be easy to come up with 1 or 2. Coming up with 3 can be a challenge. Whenever you're coming up with hypotheses, it is a good idea to write them down in complete sentences. So let me give you an example of what I mean here just verbally. Why do female honeybees mate with so many different males? Female honeybees mate with so many males because they're trying to get a lot of sperm. So the bullet point there was trying to get a lot of sperm, and the full sentence is they're doing this because of this reason. The reason that it's a good idea to write it in complete sentences is it's going to help us figure out how to design a good test later, and so we'll come back to that in a little bit. But for now, come up with 3 hypotheses. Pause this if you like. So, I don't know if I told you this, but queens, the queen honeybee, is actually mating with this male here. You can tell the difference between the male and the female because the male has a really small head and the female has a really big head. And the male honeybee is actually literally piercing the female's abdomen with his stinger-like penis when he finishes the injection process. When he pulls away, it's just like any honeybee that stings you. It actually rips their guts out from them, and they die afterwards. And the female just goes ahead and mates with another male. So that's interesting biology right there. And sorry about that. And it's going to be a little bit important for understanding what we're going to talk about soon, which is that that a couple of scientists actually came up with their own hypothesis. They decided that female honeybees mate so many times because they're trying to increase genetic variation in offspring, which makes sense. But they had this more specific idea. Remember how I told you that hypotheses are specific? So they thought that the genetic variation would actually increase the hive's resistance to parasites. So there's only one real queen in any hive, and so all of those offspring, all those bees in that hive are her offspring, and they all work together. So maybe having genetic variation will help this disease of parasites be decreased. So this was done in 2007. This was published in 2007 by 2 people, Seeley and Tarpey. And what they did to do this was they artificially inseminated the bees. Now, when I start talking to you about artificially inseminating bees, we're actually starting to get into designing a test. And so this was what they decided to do as part of their design. But it turns out that designing a test is one of the more challenging aspects of coming up with a good scientific test, like the whole process. Designing the test is one of the more challenging parts. So it's not, I don't want you to just think, oh yeah, well, okay, they artificially inseminated this. It turns out it's pretty easy to do that. You need to get the sperm out of the male, and you just inject it into the female like the male would have done. So it's actually pretty easy to do. But when we're thinking about designing a test, you don't just think about that one little aspect, that key thing of what you need to do. There's actually several things that I'm going to be asking you to think about. And here's 3 of them. And I'm plopping them up here on the screen all at once because they're really related, and I want you to be thinking about them kind of all at the same time. So all of these things listed on this page are called, they're different types of variables. And variables are things that change, right? So we have our independent variable and our dependent variable. You might be familiar with them. There's some different ways of thinking about independent variables and dependent variables. But let's start with the independent. The independent variable is defined as some measurable thing that you are able to change, or whose value can be changed at the beginning, at the start of the process. So when you're thinking about causal questions, the independent variable is often the cause in the causal question that you're asking about, or it's actually like stated in the hypothesis. So that's like the independent variable. The dependent variable is like the effect. The dependent variable is created by the process of doing this experiment or making these observations, and that you as the scientist cannot control what that value that the independent variable is showing up as. You can't control that. So that's the effect of the experiment. So those two are really closely related. The control variables, notice that it's plural, control variables, those are anything that you actually want to keep the same no matter what group you're talking about. So let's go through and talk about our independent and dependent variables. It'll make the control variables make a little bit more sense. So the independent variable is this measurable entity that's changed at the start of a process. And remember, we're talking about our honeybees and why they mate so many times. So the thing that you as a scientist would have control over is you would have control over how many times they mate when you're doing this artificial insemination. And that's what Celia and Tarpey did. So their independent variable was the number of times mated, and they had 2 groups. They had a group that only mated once, and mated right in this artificial insemination kind of way. They were only inseminated by 1 male. And they had another group that was inseminated 10 times. That's the thing, that's the independent variable, the number of times that the bees mate. That's what you have control over as the scientist. The dependent variable is the effect, right? It's being created in the process. You can't control what happens, you can just observe what happens. And so what we're looking for here is parasites, right? Remember, our whole thing was, the whole possible explanation was that that the number of times mating affects the number of parasites. So the number of times mating is our cause, and the number of parasites that we see at the end of our experiment is our effect. So now we can think a little bit about the control variables. Control variables are variables that you want to be the same no matter which group you're testing. So in our bee experiment, you got the group that's only being inseminated once, and we got the group that's being inseminated 10 times. What things would you want to be exactly the same between them? Well, maybe you want them to be living in very similar circumstances, like their environment, the temperature, the amount of water they get, the amount of sunshine they get, the type of housing you keep them in, like those man-made hives, right? Those kinds of things. Maybe you want them, all the groups, to be similar in that maybe, remember, part of our hypothesis was that it increases genetic variation. Well, if we want to have a good control group control variable thing here, we would want the females to not be introducing genetic variation. We would want them to be genetically related. That way, when we bring in the males, they're the only things that are introducing the variation. There are tons of things that are control variables. The more control variables you consider, the better your experiment is designed. So besides thinking about variables, you also have to think about a control group. And all experiments, I asked the question here, do all experiments have control groups? And the answer is no. But what is a control group? It is a group that is not subjected to the manipulation, not exposed to the independent variable. Remember that our independent variable is the number of times mated. If we had 0 matings, then they would have 0 offspring, and that wouldn't be a good control group. But remember that honeybees naturally mate multiple times. They're mating 10 or more times. So a good control group for this experiment would be to have what I've described to you before as having a female only mating once. And so that is the experimental group versus the control group. The experimental group is the mating 10 times, and the control group is mating 1 time. So you don't need to take notes on this aspect of the lecture here, but I did want to tell you so you could kind of hear the independent variables, the dependent variables, the control variables, and the control group, all in the design of what Seeley and Tarpey did. So they took 35 virgin female queens that were full sisters. That full sisters is part of the control variables of genetics, right? And they were inseminated with sperm from a single male of one of 12 different hives. Remember, we want it to have genetic diversity, so we have to have 12 different hives feeding into this diversity. But here's the thing. For the first group, this is the control group, we only have 1 insemination. And those are called SDI, or single drone insemination. Drones are the name for the males. Remember, they have those small heads, so all they can do is fly and inseminate queens, and then they die. So single drone insemination. And then they did the same thing, 35 virgin female queens, full sisters to each other, also full sisters to these ones. And they were inseminated with sperm from 10 different males, from the same 12 hives that these ones were, but these ones got 10 times as much sperm, and they also got 10 times as much genetic diversity. So each of these queens were allowed to establish separate colonies in those man-made hives, and then after a while, after they had established these good hives, nice healthy hives, they were then exposed to a parasite. And that parasite is called AFB. That stands for Apian. Apian means bees. Apian foul brood. Again, you don't need to take notes on this part. I'm just kind of describing to you what was done. And then the scientists then came back 5 weeks later, which they called measurement 1, and then they came back 9 weeks later and measurement 2, just to count up how many parasites were in the colonies, and we're going to compare these colonies to these colonies. So step 5 is to make predictions, and here's what you need to know about predictions. Predictions state what will happen to the dependent variable about a specific test. And here's the key here for this good prediction. You're going to assume the hypothesis will be supported. Your book goes into a lot of details about null hypotheses and so forth, but we are not going to be really addressing the null hypothesis. It's interesting and important. We're just going to say for now that you're going to assume the hypothesis will be supported. So predictions are all about the dependent variables. You need to say what's going to happen to the dependent variable. It's a good thing we identified what the dependent variable was earlier. Remember we said that the dependent variable was about the number of parasites that we expected to happen. And so when we have predictions, we use these if-then statements. And the way that we do it is we say if, and then you go ahead and repeat your hypothesis. And then you go then, and then you state what's going to happen to the dependent variable. So let's do this together, and it might be helpful to write this down. Let's see, what was that hypothesis again? Maybe you might need to rewind or look back in your notes to think about that hypothesis. But basically, the hypothesis was all about meeting multiple times in order to increase the resistance to parasites. So here's what we would say. If mating multiple times increases genetic variation in offspring, which affects hives' resistance to parasites, that's the long hypothesis, then the hive that has been mated multiply will have fewer parasites, in other words, more resistance, and the hive that has mated singly will have more parasites. So that's your if-then statement. Remember that you have to talk about the specific test in there. But when you're talking about the specific test, it's in the then part. You have to say what will happen to the dependent variable in that test. And the if part is only repeating the hypothesis. A classic mistake that students make is they say, if I expose them to parasites, that's not the hypothesis. You have to restate or repeat the actual hypothesis that you did. So be real careful there. So step 6 is gathering the results. And here you're actually performing the test that we've already described. But really gathering the results is all about presenting results. And you can present these either in charts or in tables or in graphs. But the thing about presenting your results is that they tell about the trend of the data. They tend to talk about averages. They tend to talk about, well, when you're presenting the results, you're not going to talk about every single one of those 70 bees. You're going to say, oh, the bees that were in this control group versus this experimental group. Also, the thing about results is that you really don't state your opinions when you're making a graph. You literally just make the graph. And so a lot of people want to say, oh, look at this data are really good. That's the last step. That's drawing the conclusion. This step we just show about trends. So there's lots of different ways that you can talk about trends. You can show about trends in graphs, and that's by thinking about the different graphs. So I've grabbed this from your textbook, talking about the different kinds of graphs that you might see in the textbook. There are other kinds of graphs. We'll talk maybe in lab about a line graph that has, instead of connect the dots, maybe we'll do a best fit line. But for now, they're just these basic ones. So the bar graph. The bar graph has the rectangular bars, and the height represents the proportion of how many things fit into that. A line graph sometimes connects the dots like you see here, or again, maybe we might see a trend line instead. We do talk about that in class. This just kind of shows what happens across many data points. And then the last one is the pie chart. Pie charts have they have slices that are each proportionally different sizes, and so it's like each slice of the pie. If it's a small slice, that means that is less of the whole than a larger slice of the pie. We do actually use pie charts in class, and they're very useful for what they are. So let's go ahead and take a look at, if you're making a graph, what you should include. And here's where your book and I actually disagree a little bit. Your book says you should have a title. I'm not fond of titles. on graphs at all, and so I never require that you have a title. Instead, what I require is that you have really well labeled axes. So I've got this little cartoon here telling you that you should always label your axes. Those are axes, but axes is plural for axis. So this is the y-axis. That's the vertical axis, and that is often that's going to be your dependent variable on the y-axis. It's not always true, and your book does mix it up a little bit, like sometimes they turn graphs on their sides, and so the vertical axis is not the dependent variable. But remember, as long as you know what a dependent variable is, then you can recognize which one you're looking at. So that's really important. So y-axis tends to be vertical and the dependent variable. The x-axis, that's the horizontal axis, also has a really good label. Notice that if you look at the x-axis and the y-axis, you actually come up with the title. So this axis is time spent studying each day, how many in hours, so 0 to 5 hours spent each day. And this is the y-axis is the dependent variable, performance on midterm exams, and that's in percentages, so it tells you the units. And you can see that in this particular study, people who studied 0 hours each day were scoring pretty poorly on the midterm exam, and people that studied more each day were doing relatively more. It also, interestingly, at least from these data, show that just an hour a day is pretty good. Like that really bumps you up from here getting a D to getting an A. So maybe that's a lesson that you can learn for yourself too. But the thing that I'm trying to get to is that I want you to label your axes really carefully with descriptions as well as units. Independent variable, dependent variable, cause, effect. So now that we've talked about the general aspects of graphs, we're going to talk about the data or the results from the honeybee study. Now, this graph right here, the second you look at it, it's going to look overwhelming. But remember what we just went through. We just broke down what a graph was trying to teach you. And the first thing I want you to ask is, what kind of graph is this? And it's got rectangles, right? That makes it a bar graph. And it's a schmancy kind of bar graph, but that's all it is a bar graph. So don't worry about all of the other details. Just focus on the rectangle sizes. The next thing I taught you is to look at the axes and notice that the axes here are really well described. It tells you that we're looking at AFB intensity. Remember, AFB is the name of the apian foul brood. That's the name of the parasite. And notice also, it doesn't just leave it at that. It actually says, hey, that's infected larva per full frame of brood. It actually gives you information about how they collected it. We have measurement 1 right here, and we have measurement 2 right here. So this graph is about measurement 1. That was how many parasites after 5 weeks. And this one is measurement 2. That was how many parasites after 9 weeks. So if we now look at the x-axis, we can see that the ones on this side are the multiple drone inseminated. They got injected 10 times. And these are the single drone insemination. And then we have the overall insemination treatment. So that actually makes, when you start paying attention to the details, that makes reading this graph so much easier. So we can compare multiple drone insemination versus single at 5 weeks. Hey, look at that. There are more intense infection in the single drone inseminated bees. And 4 weeks later, you can see the number of parasites definitely picked up in the multiple drone insemination, but check it out, it really increased in the single drone insemination. Now remember, when we're talking about the results, we don't give our opinion. So at this point, we're not going to give our opinion. I've just described this graph to you, and that's key. Let's do a little practice here, just taking a step away from the honeybee experiment and think about this graph right here. Look, notice there's no title, but they do have axes, and this little creature here kind of looks like a chipmunk, or oops, oh dear, a chipmunk or something. And so what are the axes? What kind of question were the scientists asking? So we've got number of survivors on the y-axis, that's the dependent variable, and we have age on the x-axis. So maybe the researchers were asking how many survivors, or how long do individuals of the species survive over time. Or maybe they were more interested in male versus female, like which one lives longer, males or females? So both of those are good possible questions that they were asking. Here's another one. Also, this one's also about grades. Grabbed it off the internet, so bear with me. It has a title, and then it has class average. Now, class average, it doesn't have a unit, but it does have a list here, and you might assume that that's percentage, and I think that's a pretty good assumption. And the x-axis is days absent, so the graph kind of tells you what kinds of questions or hypotheses even that they were asking. I think that this person was asking, how do missed days affect the average for individual students? And the red line is the class average. So the more days that were missed, the lower the average is for those people who missed that many days. And also an important lesson maybe to learn for our class. We do take attendance, so just kind of be aware of that. The last step in the 7-step process is to generate a conclusion, and a conclusion is where you get to say your opinion. It determines whether or not the results support or provide evidence for or against the hypothesis. So thinking back to the honeybee experiment, we are going to talk about the conclusion, but remember that this is actually where we're interpreting the results. Do you think that the results support the hypothesis or not? It's really important when you come up with a conclusion that you don't restate the results. Remember, the results for the honeybees were really factual, like the single drone insemination had more parasites. That's a result. We don't want to restate the results here. We want to say our opinion about those results. And so we have to relate back to the hypothesis. And we have to say whether or not we think the data are supporting that hypothesis or rejecting it. So here we go. What should the conclusion be? And you can pause this to write it down. I have it on the next slide if you're impatient. But let's go ahead and look at that version of it that I have. And basically, remember here it says we want the hypothesis, then you have to repeat the hypothesis. And you have to say whether it was supported or rejected. So right here we see the hypothesis that multiple mating in honeybee queens affects colony resistance to parasites was supported. The hypothesis that, repeat the hypothesis, was supported or rejected, pick one. The hypothesis that multiple mating in honeybee queens affects colony resistance to parasites, that's the first part. And I say it was supported. I think the data are pretty clear about that. They're really clear. I mean, these are some really good data. Why not be more firm? Why not say that the evidence proves the hypothesis? I really, really don't like it when students use the word prove for lots of good reasons. For example, if you're a scientist and you're saying, well, this proves my hypothesis, maybe somebody comes along later and says, well, but your scientific experiment wasn't designed that great. Or anytime you say the word prove, you're like literally shutting down curiosity because somebody proved it, right? And I want you to remember that from the beginning of this lecture, we started talking about how science is curious, and curiosity is really important. So don't use the word prove. If you think a hypothesis was supported, say it was supported. If you think it wasn't supported, you can say not supported, or you can say rejected. Either one of those is fine. I'm going to go ahead and pause this and start up a new recording because it's been about 30 minutes.